Eugene Pergament. Eugene is Emeritus Professor of Obstetrics and Gynecology at Northwestern Medical School, President of Northwestern Reproductive Genetics, etc. He was instrumental in first introducing in the U.S. number of significant approaches to prenatal diagnosis, including PGD, first trimester chorionic villus sampling, and combined risk assessment for aneuploidy based on age, serum markers, and fetal ultrasound. And personally, Eugene has been very helpful to me because he helped me obtain my first fellowship. So I'm forever grateful to Eugene.
I sent him somewhere else.
You did send me somewhere else. And Eugene will discuss the origin of aneuploidy, which I think is very important for us, both in prenatal diagnosis and pre-implantation genetic diagnosis. And it's the question that remains unresolved.
Yes. Why study the origin of aneuploidy? It's now almost 6 generations, 6 decades, I should say, in which the first major aneuploid conditions, trisomies 13, 18, and 21, as well as the sex chromosomes were described. And yet in that 60 years, we have not come to agreement as to the origin of these aneuploidies. So why study them? Well, the anthropologist Ashley Montague was quoted as saying, the purpose of science is to understand nature and then to control it. And this origin of aneuploidy would be an excellent example of, if we had understanding of its origin, we may be able to implement various strategies to reduce their incidence or eliminate them if possible. So I want to discuss with you the origin of aneuploidy from 3 levels. They're artificial levels, to be sure, but I thought they would compartmentalize our thinking as to the nature of possible examples or possible explanations as to the origin of aneuploidy. At the chromosomal level, there are 3 major mechanisms that have been identified as contributing to the origin of aneuploidy. And the largest category, about 90% of the cases, involve premature separation of sister chromatids, and is illustrated here where one of the chromosomes, the chromatids separate prematurely, if you will, one being included into the polar body and the other one being retained in the maternal egg. The original thinking was that the major sources of aneuploidy were due to nondisjunction, in which the entire chromosome, both chromatids, were either excluded or included in the polar body or the egg. And it turns out that that only constitutes about 1 or 2%. So we have to be careful of our language, because many times people describe these chromosome abnormalities as arising by nondisjunction. And if you want to use it as a general term, that may be agreeable, but if you want to be very specific, nondisjunction actually forms only a very small part of the etiology leading to aneuploidy. And the third category, I don't know if you can see it if I'm not blocking it, has been recently described and emphasized by one of our colleagues in the audience. Alan Handyside is here, of reverse segregation, in which each of the chromatids separate from one another, and so you do not have the underlying basis of meiosis, and that is to separate homologous chromosomes from one another. And in reverse segregation, this basically failed because each chromosome has separation of its chromatids, and that will cause problems primarily in the meiosis II in the second generation, where these chromatids will act as independent agents and may be included or not included in the subsequent products of forming. There are two major explanations for the origin of aneuploidy. One involves the cohesion complex, and the other kinetochore-microtubule interactions. The cohesion complex is actually a simplified term for a series of biochemical actions and reactions, as is true for the kinetochore microtubules. These are simplified statements, but these are extremely complex constellations of chemical reactions that are taking place. And despite the fact that for 50, 60 years we have had knowledge of these two particular entities, there is still, as one reads the literature, a great deal of differences as regard to the relative importance of these two systems contributing to aneuploidy. And I'll try to illustrate that to you. This is an illustration. I think I can use this here.
It's hard to,
Yeah.
it doesn't show well on the screen.
Okay. So I'll yell. Generally speaking, we talk about cohesion holding the chromatids of a single chromosome together, and the kinetochore as the basis for chromosome movement and separation. This is an example on the right of a single cohesion entity, which is a circular structure in which one consisting of 4 subunits. Rec8 is the most important one because that's the entrance and exit, primarily exit of chromosomes. And this illustrates the two chromatids being held together by this circular structure of cohesion. So automatically you see that for each of these subunits of the cohesion molecule, you have a whole series of biochemical reactions contributing to their formation and their function. And basically what we see on the left-hand side in a young bivalent is cohesion maintaining a connection between chromatids as well as between chromosomes. Kinetochores holding the chromatids together from a single chromosome. As these bivalents age, you'll notice two major occurrences. One, a reduction in the amount of cohesion, both in terms of the distal as well as particularly toward the centromere. And you will notice that the distance, and this has been measured, the distance between the sister kinetochores separate. And that separation has been used as one major explanation for explaining the age-related effect of certain chromosome abnormalities, such as trisomy 21. This is an example of the reverse segregation, which is showing that the chromosome has been altered in such a way that the kinetochores of a single chromosome, the two chromatids are going in opposite directions to different poles. And basically what underlies meiosis is the separation of homologous chromosomes. And this, in a sense, represents a violation of that paradigm whereby instead of the chromatids moving in the same direction, in unipolar direction, they move in opposite direction and set up this phenomenon called reverse segregation. And that's not been fully appreciated. I'm listing for a particular purpose the different functions of cohesion. First of all, I've indicated that they maintain a connection between sister chromatids. They are involved in recombination, in the pairing of chromosomes, in the assembly of the synaptonemal complex, the complex that leads to crossing over. So we have apparently attributed a whole series of very distinct functions to this cohesion. And that, to my mind, is one of the troubling thoughts, at least I have, about the role of cohesion in terms of causing nondisjunction, loss of the maintenance of the proper chromosome distribution. And toward the end, I'll give you an alternative that may show that there are other components in this biological system contributing to disjunction and non-disjunction. This is a series of titles of papers that I thought I would simply read one or two of them to illustrate the very diverse functions attributed to cohesion. And in light of the cohesion-related causes of aneuploidy, weakened centromere cohesion, premature dyad separation in meiosis II, that's early separation of the chromosomes in meiosis II, exhaustion of cohesion, depletion of cohesion, SMC1 beta deficiency, which is another component of the ring of cohesion, premature separase activation, which is responsible for maintaining the ring of cohesion. And so you see that there are a number of phenomena that have been biological phenomena that have been described for cohesion. And it's, again, to my mind, troublesome that one unit has been given so many responsibilities. I'm not sure in general biology works that way. The same thing is true for the kinetochore in terms of dysfunction. And there are a whole series of very important papers with very defined rules as to how they have applied their analysis, which indicates that in many instances it is the kinetochore and the dysfunction of the KINETOCHORE-MICROTUBULES that's contributing to aneuploidy. And I think it begins to arise in our minds, is there a unified, simple explanation for aneuploidy? And this is beginning to tell us if all of these factors involving cohesion and the kinetochore are really valid, because most of these experiments are artificial. We are not looking at a natural system in many instances. We create the conditions that test for cohesion. We create the conditions for the kinetochore. So we are artificially intruding into the natural biology, and that has never been articulated as a major source of criticism, but I think a valid one at that. Meiotic recombination, where the chromatids are exchanging genetic material, has also been attributed as a major third factor in contributing to aneuploidy. So we have at least two explanations now: alterations in the behavior of cohesion in the KINETOCHORE-MICROTUBULES, as well as in meiotic recombination. I introduce now two relatively new topics with regard to aneuploidy. And I say they're new because they first were described in January of this year, in back-to-back issues of Science that I've cited. And one is the protosome, which actually is a biological system that in general recognizes when chromosomes that are not paired properly. It recognizes this failure of chromosomes to pair properly, and stops that kind of synapsis process from occurring, and replacing it in finding the proper homologue to pair as part of the essential part of meiosis. And I'm sitting and thinking, how do these chromosomes think, if that's not my member, my mate, so to say, I have to look around. I have to literally move from chromosome to chromosome until I find the How is that taking place? It almost sounds a little bit like these chromosomes have a mind of their own, if you will, and it's being expressed in terms of meiosis. And the fifth player described in science is Actin, in which it protects the egg against chromosome segregation errors. And this is a paper by Mogisi and Shu in Science, again, January of 2017. So we are faced with a whole series of different biological systems, like constellations in the universe, all interacting, circling around one another. Actin apparently promotes chromosome segregation in female meiosis, and any changes in actin apparently leads to errors in chromosome segregation. And notice the different places where actin can have a role in metaphase 1, anaphase 1, metaphase 2, anaphase 2. And I've listed there some of the consequences of the failure of actin to perform properly. So finally, to the question, what in vivo process fails in meiosis, I've sort of listed them as a sort of a stairwell or stairway, starting with the protosome, keep the homologs together, if you will, cohesion, keeping the chromatids in place, recombination, the kinetochore, spindle assembly, actin, all contributing to producing, presumably, a euploid. And apparently, errors can take place in any one of these systems. A colleague of mine, Jolie Simpson, once made a comment, given all of this, it's a miracle that any one of us are in this room talking to one another. I'll stop there.